Welcome to the EvoEd COVID-19 video series. In this video, we'll take a look at how vaccines provide hosts with immunity against SARS-CoV-2. By the end of this video, you should be able to describe how COVID-19 vaccines trigger an adaptive immune response to protect hosts against the virus, and compare and contrast the mechanisms between mRNA vaccines and viral vector vaccines. First, let's take a look at the novel mRNA vaccines used to protect against SARS-CoV-2. These vaccines consist of a lipid nanoparticle containing pieces of mRNA that code for the SARS-CoV-2 spike protein. Similar to the cell membrane, lipid nanoparticles consist of a phospholipid bilayer, allowing them to easily enter the cell through endocytosis. Once the mRNA is inside the cell, it is translated by the cell's ribosomes into SARS-CoV-2 spike proteins. Innate immune cells, such as dendritic cells, present these spike proteins to B cells, which begin the production of antibodies that can bind to the spike protein of SARS-CoV-2 viruses. Memory B cells retain these antibodies and remain ready to trigger an adaptive immune response if these antibodies detect SARS-CoV-2 viruses in the host. This training of the adaptive immune system allows for a rapid immune response if infected, and greatly reduces the probability of severe disease. Now let's take a look at viral vector vaccines. These vaccines consist of a deactivated adenovirus with a double-stranded DNA molecule that codes for the spike protein of SARS-CoV-2. This virus binds to receptors on cells, and similar to lipid nanoparticles, it enters via endocytosis. Once inside the cell, it sends its DNA into the cell's nucleus through nuclear pores, where it is transcribed into mRNA. This mRNA travels back into the cytoplasm, where it is translated into spike proteins. Once these spike proteins are produced, they can be presented to B cells, which produce SARS-CoV-2 antibodies, ultimately resulting in the same outcome as mRNA vaccines. When comparing mRNA vaccines to viral vector vaccines, in terms of similarities, both work by getting mRNA into human cells and ultimately producing spike proteins that train the adaptive immune system to produce antibodies against the virus. They're also similar in the sense that they're both effective and can be developed and produced quite rapidly. And in terms of differences, mRNA vaccines enter the cell in a lipid nanoparticle, where the viral vector vaccines enter through an adenovirus. And also in mRNA vaccines, the mRNA is directly translated once it's in the cytoplasm, where in viral vector vaccines, they first must have their DNA transcribed into mRNA, which can then be translated into the spike proteins. So that's it for this one, and that concludes our COVID-19 video series. For more interactive case studies, please be sure to visit our YouTube channel and also our website at evo-ed.org. Thanks for watching.